Cell cycle and cell division. Significance of cell reproduction. All cells are formed by the division of pre-existing cells. Each new individual will begin its life as a new single cell, commonly the fertilized egg or zygote by the cell division. A large number of cells are being torn or killed every moment in the body of a multicellular organism through either skin peeling, lining of digestive tract, destruction of old red blood cells or capsules in an adult human body. They are being continuously replaced through the formation of a new cell. An injury is healed through the formation of new cell by healthy cell around the area of injury. It is the mode of multiplication in unicellular organism. In multicellular organism, cell reproduction is required to form propagules or gametes. The mechanism of cell reproduction or cell division is fundamentally similar in all organisms, showing the kinship or unity of life. This is the significance of the cell reproduction. Then we have factor controlling the cell reproduction. A number of factors are known to induce the cell division. They are minimum growth. A newly formed cell does not divide immediately. Some amount of minimum growth in the cell and its component is required before a cell attains the ability to undergo division. Then we have surface volume ratio. Increase in the cell size will result in decrease of the surface volume ratio. This disturbs the efficiency of surface exchange required for maintaining optimum metabolism. As it reaches a critical stage, the cell undergoes division. Nucleocytoplasmic or Kern plasma ratio. Cell functions are controlled by nucleus. The size of nucleus will not change while that of a cytoplasm will increase during the cell growth. As nucleocytoplasmic ratio decreases, the cell is stimulated to divide. Mitogens, they are substances or factors which bring about the cell division. Cytokinin is a plant hormone which functions as a mitogen. Other mitogenic substances in human being are EGF or PDGF and lymphokines. There are some agents which inhibit cell division. They are called mitotic poison. For example, azides, cyanides, chalones, and colchicines. Colchicines is obtained from the etu- um, Echococcus. It arrests cell division at metaphase due to non-formation of spindle. Let's look at cell cycle. Cell cycle is genetically controlled series of changes that occurs in newly formed cell by which it supplicates its content, undergoes growth and division to form two daughter cells. It consists of two states or periods: a long non-dividing growth phase, which is your I phase, interphase, and a short dividing M phase. Both have substages. I phase represent interphase. The regular sequences of G1, S1, and G2 is the interphase. And M phase is mitotic phase and it is called the cell cycle. Interphase is also known as the resting stage, but it is in fact a period of great activity. Three important processes which are preparatory to cell division take place during the interphase. These are replication of DNA along with synthesis of nuclear proteins such as histones. In element cell, duplication of centriole take place by outer growth of daughter centriole from the parent centriole, which are at right angle to each other. Synthesis of embryo, rich compounds which provide energy for mitosis, and synthesis of protein at the end of the interphase. Interphase is intermitotic stage of cell division in which series of changes occur in newly formed cell and nucleus undergoes certain changes to be fixed for the division. Non-dividing state of mature cell or nucleus is called interphase. It is also called the energy phase. Interphase of dividing cell has been classified into the three subphases as we discussed earlier: G1 phase, S phase, and G2 phase. Here you can see the diagram of G1 phase, then we have S phase and G2 phase, then we have M phase, which is further divided into prophase, metaphase, anaphase, telophase, and cytokinesis. What happens in G1 phase? G1 phase is also known as the first growth phase or post-mitotic gap phase. It is the longest phase of the cell division. In this phase, different type of RNA, for example mRNA, tRNA, rRNA, and protein are synthesized. All cell organelles, mitochondria, ER is endoplasmic reticulum, mitochondria, Golgi complex, ribosomes, plastids multiply. The duration of G1 phase varies from cell to cell. It is shorter in frequently dividing cell. G1 phase cell has three options. A. Continue cycle and enter S phase. B. Stop cell cycle and enter quiescent phase or G0 phase. C. Stop cell cycle and undergoes cell differentiation. The deciding factor for above options are availability of mitogens, an energy-rich compound. These point is called checkpoints. Once the checkpoint of G1 phase is crossed, cell reaches a state called antiphase, whereby it will divide even under unfavorable conditions. Cell cycle will go on further division till completion. Then we have S phase. S phase is known as synthetic phase. In this stage, replication of DNA takes place by synthesis of histones. As a result, each chromosome undergoes replication, producing two chromatids. Each chromosome will carry a duplicate set of genes. Haploid cell N becomes diploid 2N, which means one cell gives rise to two daughter cells, and a diploid cells 2N, two daughter cells um, thus become tetraploid 4 cells at the end of S phase. Repairing of damaged DNA also take place. Then we have G2 phase. G2 is also called second growth phase or pre-mitotic gap phase. In this phase, synthesis of DNA stops and synthesis of RNAs and protein continues. All cell organelles multiply and spindle formation take place. It lasts for 2 to 5 hours in most cycles. Some protein formed in this phase cause condensation of chromosome to initiate mitosis. G0 phase, the phase in which cell fails to divide further and do not undergo S phase after G1 phase and undergoes differentiation, is also known as G0 phase or quiescent stage. It occurs due to non-availability of mitogen, an energy-rich compound. The cell remain metabolically active, grow in size and differentiate for particular function after attaining a particular shape. However, some cells remain in undifferentiated state as reserve cells. They may proceed with cell division when required. For example, fibroblasts, it will help in healing of wounds and grow and divide again. M phase, mitotic phase, the process of cell division is found to be essentially the same in all living organisms and events are chiefly centered in the nucleus. Three types of cell division have been distinguished. Amitosis or a direct cell division, mitosis, indirect cell division, and meiosis, a reduction division. Mitosis and meiosis are the two major types of cell division. The basic stage in both types of division are almost identical. Amitosis is a different division characterized by splitting of nucleus followed by the cytoplasm. Mitosis is somatic cell division which takes place in vegetative cell. It maintains the chromosome number. Meiosis is a reduction division occurring in the reproductive cell. The chromosome number are reduced to half. Let's look at amitosis. Amitosis is a method of direct cell division in which nucleus constrict into two daughters without showing differentiation of chromosomes and development of spindle. Nuclear division is followed by cytokinesis. Cytokinesis is your division of cytoplasm. Amitosis was first described by Robert Freeman in red blood corpuscles of chick embryo. The term was coined by Fleming. Occurrence It occurs through the cleavage or constriction of, example, cartilage cell, degenerate cells, meganucleus of paramecium, cells of fetal membrane of vertebra. Moneron cell division is sometimes included under amitosis due to absence of spindle. Its drawback, as amitosis does not distribute chromatin equitably, it results in structural and functional abnormalities in cells. Then we have mitosis. It is a cell division in which chromosomes of parent cells are duplicated into two daughter nuclei. The term mitosis is derived from the Greek word mitos, which means thread. Mitosis was first observed by the Strasburger in plant cells and Boveri and Fleming in animal cells. The term was coined by Fleming. It is also known as equational division due to equal distribution of chromosomes in daughter nuclei. It is often known as somatic cell division due to occurrence in somatic cells, 1 to 5% of total duration of cell cycle. On the basis of different type of cells and species, mitosis takes place about 30 minutes to 3 hours for completion. In plant, all mesosomatic regions are the site of mitosis. For example, root, ape, shoot, apex, lateral meristem, leaf, flower, fruits, embryo, etc. In animal embryo, skin, bone marrow are the site of mitosis. Mitosis is completed in two steps. One is karyokinesis, in which nucleus divides, and then we have cytokinesis, in which cytoplasm divides. Karyokinesis, mitosis starts with nuclear division of parent cells known as karyokinosis. The four phases of karyokinosis are prophase, metaphase, anaphase, and telophase. Prophase is often divided into three substages. Early prophase, mid prophase, late prophase. It is the first stage of mitosis proper. It is the longest phase of karyokinosis. In the early stage of prophase, in this substage, nucleus and cell become spheroid and nucleus appear as a ball of wool. Chromatin fiber condenses to form elongated chromosomes, and this increases the viscosity and refractivity of cytoplasm. In animal cells, duplicated centrioles start to move towards opposite pole of the cell. Each centriole will radiate out fine microtubules, microtubular fibrils called astral rays. In animal cells and cells of lower plants, fibril appears like spokes of a wheel around each centriole to form an ester. Then we have mid-prophase. In mid-prophase, chromosome shifts toward the periphery and leaves a clear central area. It becomes shorter and thicker. Each chromosome consists of two longitudinal threads called chromatid. Both chromatid are attached to each other by centromere and are known as sister chromatid. Then we have late prophase. In this substate, spindle fibers start appearing around the nucleus. The size of chromosome in which reduced or compared to early prophase. Spindle poles are formed without acids in plant cells and with esters in animal cells. Nucleolus and other cell organelles like mitochondria, Golgi complex, vacuole will disappear. The presence of spindle is essential for mitosis. If cells are treated with collikines, which inhibit spindle formation, anaphasic movement of two groups of chromosome to pore does not take place. Prometaphase, pro mean before. So before metaphase, there is a stage known as prometaphase. It is intermediate stage of prophase and metaphase and hence acts as connecting link between them. Nuclear membrane completely degenerate in this stage. So mixing of the cytoplasm with nucleoplasm occurs. It is known as extranuclear mitosis and eumitosis. In many protozoan, fungi and some animal cells, nuclear membrane does not degenerate throughout the cell division known as intranuclear or pre-mitosis. A spindle fiber consists of 4 to 20 microtubules formed of protein tubulin. Spindle fiber coverage at two ends or poles. It has maximum diameter in the middle known as equator. Then we have metaphase. In metaphase, discontinuous fibers radiate out from the two poles and get connected to the disc-shaped structure at the surface of centromere called kinetochore. A kinetochore is complex protein structure that is analogous to ring for the microtubule hook. It is point where microtubule attach themselves to chromosomes. Chromosomes or kinetochore fiber contract and bring chromosomes over equator. This phenomena is called congregation. Smaller chromosomes directed towards the center while larger ones are in the peripheral in position on the equator. The centromere of all the chromosomes lies on the equator, forming an apparent plate called metaphasic or equatorial plate, while arms are directed towards the pole. Kinetochores have two functions. Main function apparently is that they serve as the attachment of microtubule of the chromosomal spindle fiber. They might also be involved in formation of chromosomal spindle fibers during the prometaphase and metaphase by serving as center of the polymerization of proteins of microtubule. Metaphase is the best phase to count total number of chromosomes in any species and detailed study of morphology of chromosome. Then we have anaphase. Ana means up and phase is stage, so up stage. Chromosomes are arranged on the equatorial plate of a short period. The centromere of a chromosome start to divide into two, forming daughter chromosomes with centromere in each. Daughter chromosomes are repulsive, so migrate towards the opposite pole. Spindle fibers attached to the centromere shortens and pull the chromosomes towards the pole. The velocity of anaphasic movement does not depend on the size of a chromosome. In anaphasic movement of chromosomes, the centromeres lead the path while the limb trails behind. So, anaphasic chromosome, the centromere lead the path while the limb trail behind. It appears as V, L, J and I shape. Metacentric is V shape. Centromere position is in median. Submetacentric is L shape. Submedian. Ectocentric is J shape. Subterminal and telocentric is terminal centromere position. At the end of the anaphase, two groups of chromosomes are formed, one at each pole. The number and type of chromosome at each pole is the same as in the parent nuclei. Last is telophase. During telophase of mitosis, viscosity of cytoplasm decreases. A nuclear membrane is formed around each set of chromosomes. Chromosomes overlap one another forming chromatin. The nuclear organizer region of a satellite chromosome produces nucleolus for each daughter nucleus. Nucleoplasm surrounds in the area of chromatin. The gel state of spindle is converted into Sol state and disappears. In this way, two daughter nuclei are formed at the pole of spindle. Hence, this phase is just reverse of prophase. Golgi complex and endoplasmic reticulums are formed. Cytokinesis starts either by cleavage or constriction. Cytokinosis. Mitosis ends with division of cytoplasm and it is known as cytokinosis. It is derived from the Greek word cytos means hollow or cell mean kinesis. Kinesis means movement. It starts towards the middle of anaphase and is completed with the telophase. It is different in animal and plant. If nuclear division takes place without cytoplasmic division, and syncytium is formed. Animal cytokinesis, the spindle gets charged into dense fibrous and vesicular structure on equator called mid-body. In the middle region of the cell, microfilaments start to be collected, which induces the cell membrane to be invigilate. The furrow forms and deepens centripetally and finally cleaves the parent cell into two daughter cells. This method of cytokinesis is known as cleavage method. All cell organelles are also distributed between two daughter cells. Then we have plant cytokinesis. It differs from the animal cytokinesis due to presence of rigid cell wall. In lower plant cytokinesis, occurs by cleavage method, and in higher plant it takes place by the cell plate method. In this method, small vesicle of Golgi complexes are collected at the equator, where spindle persists for some time called phlegmoplast. All vesicles fuse to form two sheets with encloses a matrix or film. This film becomes solidified to form cell plate or middle lamella. It grows centrifugally and finally phlegmoplast disappears. Cellulose, hemicelluloses, and pectin are deposited on either side of the cell plate. It forms primary wall. Here you can see in table form difference between animal and plant cytokinesis. What is the significance of mitosis or importance of mitosis? Growth. It is used for growth and development. A single cell zygote grows into full blown baby by repeated mitosis. Plants are able to grow throughout their life due to mitotic division in their apical and lateral meristems. Increase in tissue mass result from increase in cell number called hyperplasmic. Hence, mitosis is essential for growth and development of a multicellular organism. Maintenance of cell size. An overgrown somatic cells is induced to divide so that mitosis helps in maintaining a proper surface volume ratio. It is also a high nucleocytoplasmic ratio which is brought back to efficient level through division. These ratios are important for proper functioning of cell. It is also important for genetic stability. All daughter cells of a multicellular organism have same number and type of chromosomes as parent cell due to equitable distribution of all chromosomes. This helps in proper coordination among daughter cells. Healing and regeneration. For healing of wounds, new cells are produced by mitosis. Repairing. The mechanism of replacing old or worn out cells is called repairing. Evidence of basic relationship. The mechanism of mitosis are similar in majority of organisms, showing basic similarity and relationship among them. Here we have the diagram showing the different stages of mitosis. Interphase. Stage 1 include G1 phase of interphase and G2 phase of interphase. Then we have mitotic phase, starting with prophase. In G1 phase of interphase, chromosomes are unreplicated and extend throughout the nucleus. For simplicity, we show only two pairs of chromosomes. One of each pair was inherited from one parent and other was inherited from the other parent. Then we have G2 phase. This is also part of interphase. After replication during the S phase of interphase, each chromosome is double at all points and now consists of two sister chromatids. Cohesion encircle each pair of sister chromatid along their length, aligning them tightly. The centriole within centromere also have doubled into the pairs. Prophase. Chromosome condenses into thread that become visible under the light microscope. The tight alignment of the pair of sister chromatid are now be seen. The centromere has divided into two parts which are generating the spindle as they separate. You can see here sister chromatid. This is whole chromosome consisting of two sister chromatid pairs, microtubules of the developing spindle. Then we have pro-metaphase. The nuclear envelope has disappeared and spindle enters the former nuclear area. Microtubule form opposite spindle poles attached to the two kinetochores of each chromosomes. Next phase is metaphase, where chromatids line at the equator. Chromosomes become aligned at the spindle midpoint. Each sister chromatid pair is held in position by the opposite forces. The kinetochore microtubules pulling to the poles at the cohesion, binding the sister chromatid together. Next is anaphase. Separate cleaves the cohesion ring responsible for sister chromatid cohesion. The spindle separates the two sister chromatids of each chromosome and move them to the opposite spindle poles. Here you can see chromosome splits into the chromatid and they are being pulled at the poles by the kinetochore microtubules. Then we have telophase and cytokinesis. The chromosome unfold and return to interphase state and new nuclei develop envelopes form around the dotted nuclei. Cytokinesis now commence as seen by the cytoplasm begin to divide by furring at the point marked by arrow. Lastly, G1 phase of the following interphase. The two daughter cells are genetic duplicates of the parent cells that enters the mitotic division again. These are your different phases of mitosis or stages of mitosis. You can remember this by the mnemonic IP, MAT. I stands for interphase, P is for prophase, M for metaphase, then we have anaphase and finally telophase. So IP, MAT for mitosis. Regulation of mitotic cell cycle by internal control. In the diagram you can see different phases of cell cycle, which is G1, S phase, G2 phase and M phase, but these are regulated. Three key checkpoints for G1 or S transition, for G2 M transition, and for the attachment of chromosome to mitotic spindle monitors the cell cycle event to prevent crucial phases of cell cycle from starting until previous phases are completed correctly. Complexes of cyclin and cyclin dependent kinases CDKs regulate the progression of cell through the cell cycle. The three cyclin CDKs present in all eukaryotes are shown. The CDKs are present throughout the cell cycle, but they are active only when complexed with cyclin. Each cyclin is synthesized and degraded in a regulated way so that it is present only for a particular phase of the cell cycle. During that phase, the CDK to which it is bound phosphorylates and thereby regulate the activity of target protein in the cell that are involved in initiating or regulating key events of the cell cycle. Let's look at different checkpoints. Checkpoint number 1 is G1 or S. It is the main point in the cell cycle at which mechanism governing the cell cycle determine whether the cell will proceed through the rest of the cell cycle and divide. Once it passes this checkpoint, the cell is committed to continue the cell cycle through the cell division in M phase. For example, the cell cycle arrest and G1 or S checkpoint if the DNA is damaged by radiation or chemicals. G1 checkpoint or checkpoint is also the primary point at which cell reads extracellular signal for cell growth and division. Therefore, if a growth factor required for stimulating cell growth is absent, the cells are arrested at this checkpoint. Second is G2 or M checkpoint. M checkpoint is at the junction between the G2 and M phase. Passage through this checkpoint commits a cell to mitosis. Cells are arrested at M checkpoint if DNA was not replicated fully in S, or if the DNA has been damaged by radiation or chemical. The complete DNA replication is essential for producing genetically identical daughter cells, highlighting the importance of this checkpoint. Mitotic spindle checkpoint is within the M phase. Before metaphase, this checkpoint assesses whether chromosomes are attached properly to the mitotic spindle so that they are aligned correctly at the metaphase plate. The checkpoint is essential for production of genetically identical daughter cells, which depends on separation of the daughter chromosome in anaphase. In turn, the separation depends on the correct alignment of chromosome on the spindles in the metaphase. Once the cell begins anaphase, it is irreversibly committed to completing the M mitotic phase, underlining the importance of mitotic spindle checkpoint. Next we have is meiosis. Meiosis is the process of reductional division in which the number of chromosomes per cell is cut in half. In animal, meiosis always results in formation of gametes, while in other organisms it can give rise to spores. The word meiosis comes from the Greek word meon, means to make small, since it results in the reduction of the chromosome number. Meiosis is essential for sexual reproduction and therefore occurs in all eukaryotes that reproduce sexually. Meiosis does not occur in archaea or bacteria which reproduce via asexual process such as binary fusion. During meiosis, the genome of diploid germ cell, which is composed of long segments of DNA packed into chromosomes, undergo two rounds of division, resulting in the four haploid cells. Each round will produce two cells. Each of these cells contain one complete set of chromosomes or half of the genetic content of the original cell. If meiosis produces gametes, these cells must fuse during fertilization to create a new diploid cell or zygotes before any new growth can occur. Thus, the division mechanism of meiosis is a reciprocal process to joining the two genomes that occurs at the fertilization. Because the chromosome of each parent undergo genetic recombination during meiosis, each gamete and thus each zygote will have a unique genetic blueprint encoded in its DNA. Together, meiosis and fertilization consist sexually in eukaryotes and generate genetically distinct individuals in population. In lower plants and in many protists, meiosis results in formation of haploid cells that can divide vegetatively without undergoing fertilization, referred to as spores. In these groups, gametes are produced by mitosis. Biochemically, meiosis uses some of the same mechanism employed during the mitosis to accomplish the redistribution of chromosomes. There are several features unique to meiosis. Most importantly, the pairing and recombination between homologous chromosomes, which enable them to separate from each other. The cell of a particular species have a constant number of chromosomes. In sexually reproducing organisms, male and female gametes fuses together to form zygote. If gametes has the same number of chromosomes, number remain constant from generation to generation. This is because of the meiotic division, which reduces the chromosome number to half and counteracts the effect of fertilization. Thus, fertilization and meiosis are compensating events. Types of meiosis. The cell in which meiosis takes place are called myocytes. In animal, myocytes are of two types, spermatocytes or oocytes. In higher plant, myocytes are differentiated into microsporocytes and megasporocytes. Depending upon the stage when meiosis occurs, the later is of three stages: gametic, zygotic, and sporic meiosis. Gametic meiosis. Meiosis is most of the animal take place during the formation of gametes. It is termed as genetic meiosis. When two gametes fuses in fertilization, a diploid zygote is formed. Gametic meiosis results in diplontic life cycle. Zygotic meiosis. In some lower plants, meiosis takes place in zygotes and resulting organisms are haploid. It is called zygotic meiosis. Organisms having zygotic meiosis have haplotonic life cycles. Sporic meiosis. In plants, meiosis generally occurs at the time of sporogenesis, which means formation of spores or microspores or megaspores. It is called sporic meiosis or intermediate meiosis. Spores produce a new gametophyte phase in the life cycle. Gametes are formed by gametophytes because of the presence of two distinct multicellular phases which are diploid and haploid. Life cycle of a plant is diplohaplotonic. Haplontic. Phases of meiosis. Because of the meiosis is one-way process, it cannot be said to engage in a cell cycle as mitosis does. However, the preparatory steps that lead up to the meiosis are identical in pattern and name of the interface of the mitotic cell cycle. Meiosis is a type of cell division that is vital for sexual reproduction. Meiosis takes place in reproductive organs. It results in formation of gametes with half the normal chromosomes number. Therefore, haploid sperms are made in testes and haploid eggs are made in ovaries. Meiosis involves two division of the cell. These divisions are termed meiosis 1 and meiosis 2. Each one includes prophase, metaphase, anaphase, and telophase. Meiosis 1 consists of separating the pair of homologous chromosomes, each made up of two sister chromatids, into two cells. One entire haploid content of chromosomes is contained in each of the resulting daughter cells. The first meiotic division therefore reduces the ploidy of original cell by a factor of 2. Meiosis 1. In meiosis 1, homologous pair in a diploid cell separates, producing two haploid cells. So it is also referred to as a reduction division. Like mitosis, it is studied under four stages: prophase, metaphase, anaphase, and telophase. Prophase 1 is more complicated and prolonged as compared to the ciliar stage of mitosis. For the sake of convenience, prophase 1 is divided into 5 substages: leptothene, zygothene, phycothene, diplothene, and dikinases. Another subphase called pre-leptoneme is sometimes recognized prior to leptonema. In this phase, chromosomes are not distinguishable because of their thinness, but sex chromosomes are often seen as heterochromatic bodies or heterophyconotics. Leptothene. Leptothene, also known as leptonema, is the first stage of prophase 1, during which individual chromosomes begin to condense into long strands within the nucleus which are loosely interwoven. However, the two sister chromatids are still so tightly bound that they are indistinguishable from one another. Leptothene chromosomes may be irregularly arranged or may be polarized toward the centrioles, forming a bouquet. Electron microscope studies have shown that bouquet formation results when a group of chromosomes is attached close together on the nuclear membrane. In plant cells, chromosomes may sometimes form a tangle of threads called synecytic knot on one side of the nucleus. There are two sets of chromosomes in diploid cells undergoing meiosis. One set contributed by the male parent and the other by the female parent. These are always two similar chromosomes having the same size, form, and structure. They are called homologous chromosome. One of them is paternal chromosomes and other one is maternal chromosome. Next stage is zygothene. During zygothene or zygonema, the chromosomes become shorter, thicker. The homologous chromosomes come to lie side by side in pairs. This pairing of homologous chromosome is known as synapsis or syndesis. A pair of homologous chromosomes lying together is called a bivalent. The chromatids are still not visible. A fibrillar, somewhat ladder-like organelle called synaptonemal complex develops between the synapsed homologous chromosome. It is thought to stabilize the paired condition of chromosome till crossing over is completed. Pairing of two homologous chromosomes begins when their corresponding ends come together on the nuclear matrix. Pairing may occur in one of the three ways. Number one, proterminal pairing. It starts at the end and proceeds toward the middle. Procentric pairing. It begins at the centromeres and progresses towards the end. And random or intermediate pairing. It commences at many points towards the end. The synaptonemal complex is attached at both ends through its lateral elements to the inner surface of the nuclear membrane. The central element is not attached directly. Also arising from the lateral element is another series of smaller loops. These loop fuses in middle line to make up the central element. Next stage is phytene. Zygotene is followed by phytene. It is said to begin when synapsis is completed. It lasts from the completion of the synaptonemal complexes to the stage where their breakdown begins. The synapsed chromosomes continue to become shorter and thick. The chromatid of each synapse chromosome slightly separates and becomes visible. The two visible chromatids of a chromosome are referred to as dyad. A group of four homologous chromatids, two dyads, is called sister chromatids, and those of two homologous chromosomes are termed non-sister chromatids or bivalents. Crossing over recombination occurs during phytene. Recombination involves mutual exchange of corresponding segments of a non-sister chromatid of the homologous chromosome. It takes place by breakage and reunion of chromatid segments. Breakage is called nickeling. It assists by an enzyme endonuclease, and reunion termed annealing is aided by an enzyme ligase. It has been found that crossing over is a common event. Normally each tetrad undergoes at least one recombination. Next stage is diplotin. During diplotin or diplonema, the synaptic forces keeping the homologous chromosome together come to an end. The homologous chromosomes start separating. This is called disjunction. It makes chromatid more distinct and tetrad very clear. Separation of homologous chromosome does not take place at the point called chiasmata. The chiasmata makes the site where crossing over occurs during the phytene. They help in holding homologous chromosome together. Number and position of chiasmata varies with the length of chromosome and with the species. Chiasmata are found in the meiosis of the almost all eukaryotic organisms. However, achiasmatic meiosis has been reported in some organisms. For example, male of higher dipteral Panorpus or scorpion fly, many mantids and roaches, some grasshoppers and scorpions. A chiasma formed at end of a chromosome is called terminal chiasma. Chiasmata formed along the length of chromosomes are called interstitial chiasmata. During diplotin, the chiasmata begins to be displaced along the length of the chromosome. Terminal chiasma slips off the end of the chromosome and its position is taken up by the interstitial chiasma, which is now called terminal chiasma. This process is called terminalization. A diplotin progress, the number of interstitial chiasmata become lesser in number. The terminalization may be due to electrostatic forces or despiralization of the chromosome. When terminalization is completed, the homologous remain in contact through the terminal chiasma. The degree of terminalization is expressed by its coefficient. Synaptonemal complexes mostly disappear during diplotin. In certain regions, short segments may persist. The most common region where the complexes persist are near the end of the bivalent, where lateral elements are attached to the nuclear membrane, and at the site of chiasmata formation. With disappearance of the synaptonemal complexes, the axial filament become unpaired. In diploidy, the chromosomes may unfold to nearly normal form and starts transcription of mRNA and rRNA to build up the food reverse, reserves in the cytoplasm. This process is most profound in the primary oocytes of amphibians, reptiles, and birds. In some species, chromosome enlarge greatly, assuming Lambert form. Dikinesis. Dikinesis is not sharply differentiated from diploidy. The chromosomes become more contracted, the bivalents are more evenly distributed in the nucleus and migrate towards the periphery. RNA synthesis stops, nucleolus degenerates, spindle begin to develop with or without centrioles. Then we have prometaphase 1. The nuclear membrane disappears in prometaphase 1 and the chromosome reaches their maximal contraction, spindle formation begins. Metaphase 1, the chromosome now become arranged on the equator of the cell. The spindle is formed, spindle fiber become attached to the centromere of the two homologous chromosomes. The two centromeres of each bivalent lie on the opposite side of equatorial plate. The attachment of tetrate to the spindle fiber in metaphase 1 is different from the mitotic metaphase chromosome. Each homologous chromosome has two kinetochore, one for each of its two chromatid. Both the kinetochore of a homologous chromosome connect to the same spindle pole. The two kinetochore of its homologous join the opposite spindle pole. In metaphase 1 of a meiosis, there are bivalent, each bivalent consisting of the two centromeres. Then we have anaphase 1. During anaphase 1, from each tetrate, two chromatids of a chromosome move as a unit to one pole of a spindle, and remaining two chromatids of its homologue migrate to the opposite pole. Thus, homologous chromosomes of each pair rather than the chromatid of a chromosomes are separated. As a result, half of the chromosome which appear in early prophase go to each pole. It is here in the anaphase 1 that the real reduction in the pole is still double and consists of two chromatids. This is in contrast to the single-stranded chromosome in the mitotic anaphase. Paternal and maternal chromosome of each homologous pair segregate during the anaphase 1 independently of other chromosomes. Anaphase 1 is cytological event that correspond to Mendel's law of independent assortment. Although the paternal and maternal chromosomes of the homologous pair have gene for the same trait, either chromosome of a pair may carry different alleles of the same gene. Therefore, the independent assortment of homologous chromosome in anaphase 1 introduces the genetic variability. Telophase 1. During telophase 1, the chromosome at each pole of the spindle uncoil and elongate but remain straight and often do not assume the interphase form. The satellite chromosome developed forms around the chromosome and nucleoli. The spindle and astral rays gradually disappear. Cytoplasm divide at its middle by cleavage in an animal cells and by the cell plate formation in the plant cells. This produces two daughter cells. Each has received only one chromosome from each homologous pair. Thus, it has half the number of chromosomes, but double the amount of nuclear DNA as each chromosome is still double. The daughter cells formed by meiosis 1 are called secondary spermatocytes or secondary oocytes in male and female animals. Cell enter a period of rest as interkinesis or interphase 2. No DNA replication occurs during this phase. Protein and RNA synthesis may occur. It is important for bringing the true haploidy. Here you can see prophase 1, metaphase 1, anaphase 1 and telophase 1. Then we have meiosis 2. The second meiotic division is essentially similar to the mitosis. In this division, two chromatids of each chromosome separate from each other and go to the separate daughter cell. With the result, the number of chromosomes remain the same as produced by the meiosis 1. Meiosis 2 is therefore known as homotypic division. If, however, differs from the mitosis in that DNA does not duplicate, while centromere do so. It has four phases, again prophase 2, metaphase 2, anaphase 2, and telophase 2. Prophase 2 Prophase 2 take an inversely proportional time compared to telophase 1. In this process, we see disappearance of nucleoli and nuclear envelope again, as well as shortening and thickening of chromatid. Centrioles move to the polar region and are arranged by the spindle fiber. Metaphase 2 In metaphase 2, the chromosome become oriented on the equatorial plate and have a relationship to the spindles as in mitosis. Anaphase 2, where the centromeres are cleaved and also the kinetochores to pull their chromatids apart. The sister chromatids by convention are now called sister chromosomes, and they are pulled toward the opposite poles. Then we have telophase 2. In telophase 2, the group of chromosomes at each pole of a spindle get enclosed by a nuclear envelope. Nucleoli are laid swan, astral rays, and the spindles are lost. Then we have cytokinesis. Cytoplasm divide at its middle by furring in an animal cell and by the cell plate formation in a plant cell. This produces two daughter cells. The later have half the number of chromosome and half amount of nuclear DNA. These cells are mature gametes in animals and the spores in the plants. Cytokinesis may occur after each nuclear division. In such cases, it is said to be a successive type. First, the diploid parent cells divided by heterotypic division into two haploid cells, which then produces four haploid cells by homotypic division. The four daughter cells may form a linear or isobilateral tetrade. Often, cytokinesis is delayed until both the nuclear division are completed, so that the four cells are simultaneously formed, each with a haploid nucleus. The cytoplasmic division in such cases is said to be a simultaneous type. Lastly, we have the significance of meiosis. Formation of gametes. Meiosis form gametes that are essential for sexual reproduction. Genetic information. It switches on genetic information for development of gametes or gametophytes and switches off the sporophytic information. Meiosis facilitates the stable sexual reproduction without the halving of the ploidy chromosomes count. Fertilization would result in zygotes that have twice the number of chromosomes than zygotes from the previous generation. Successive generation would have an exponential increase in chromosomes count, resulting in an unwieldy genome that would cripples the reproductive fitness of the species. Most importantly, however, meiosis produces genetic variety in gametes that propagates to offspring. Recombination and independent assortment allows for a greater diversity of genotype in the population. As a system of creating diversity, meiosis allows a species to maintain stability under the environment changes. Crossing over, it introduces new combination of trait or variation. Then we have mutation. Chromosomal and genomic mutation can take place by irregularities of meiotic division. Some of these mutations are useful to organisms and are perpetuated by natural selection. Evidence of basic relationship of organisms. Details of meiosis are essentially similar in majority of organisms, showing their basic similarity and relationship. Let's look at the difference between meiosis and mitosis. In mitosis interphase, each chromosome replicates during the S phase of interphase. The result is two genetically identical sister chromatids. In meiosis interphase, chromosome not yet visible, but DNA has been duplicated or replicated. Prophase of mitosis, each of the duplicated chromosomes appear as two identical or equal sister chromatids. The mitotic spindle begins to form, chromosome condenses and thickens. In prophase of meiosis, prophase 1, crossing over and recombination, Homologous chromosomes appear together as pair. Tetrade or bivalent is the structure that is formed. Segments of chromosomes are exchanged between non-cystochromatid at crossover point known as chiasmata or crossing over. Metaphase of meiosis or mitosis. The chromosomes assemble at the equator at the metaphase plate. Meiosis. Metaphase 1, chromosome adjust on the metaphase plate. Chromosomes are still intact and arranged as the pair of homologue. Anaphase of mitosis. Anaphase, the spindle fiber begin to contract. This start to pull the sister chromatid apart. At the end of an anaphase, a complete set of daughter chromosome is found on each pole. In anaphase 1 of meiosis, sister chromatids stay intact, however, homologous chromosomes drift to the opposite or reverse pole. Then we have mode of reproduction. In mitosis, mode of reproduction is asexual, while in meiosis it is sexual. Occurrence, mitosis occur in all the cells, while meiosis only occur in reproductive cells. Function, mitosis function is growth and repair and cell reproduction, while meiosis functions as genetic, it creates a genetic diversity through sexual reproduction. Cytokinesis, it occurs during telophase in mitosis, and cytokinesis in meiosis occurs in telophase 1 and then in telophase 2. Mitosis was discovered by Walter Fleming, while meiosis was discovered by Oscar Hertwig. This is the diagram from the book. It shows prophase 1, all the different stages in the prophase 1, starting with condensation of chromosome, synapses, crossing over, prometaphase 1. Then we have second mitotic division where prophase 2, prometaphase 2 occurs. And then there is metaphase 1, anaphase 1, telophase 1, and then metaphase, anaphase, and telophase 2. This brings us to the end of unit, cell division and cell cycle.